Hello, everybody. I'm back. Dr. K.K. Agarwal here. Hello, everybody. I'm back. Dr. K.K. Agarwal, President, Confederation of Medical Association of Asia and Oceania, past National President, Indian Medical Association, and President, Heart Care Foundation of India. And you are watching Med Talks with Dr. KK, non-stop 7 p.m. show, 230th episode, and 1832nd COVID episode. Let's have the headlines of the day. The Bharat Biotech's vaccine has shown an interim vaccine efficacy of 81% in the late stage clinical trials. To set up the pace of vaccination against COVID, the government has allowed 24/7 vaccinations at hospitals with surplus capacity and resources. That means they can now do it 24/7, the whole day. Britain's medical regulator has said that it would fast-track vaccine for coronavirus variants, adding that the makers of already authorized shots would not need new lengthy clinical trials to prove their adapted vaccine will work. Research by South African scientists suggests that antibodies triggered by exposure to the country's dominant COVID variant can prevent infection by other variants. The finding in lab studies offer hope that vaccine based on the 501YV2 variant could protect against multiple variants circulating in different parts of the world. Germany will soon authorize the AstraZeneca vaccine against COVID-19 for people above the age of 65. Let's have the topic of the day. We are revisiting virus mutation every month. We have this subject so that we are updated about the latest virus mutations. Before I introduce you to the speaker, let's do a hand wash. S for Seedha, U for Ulta, M for Mutti, A for Angutha, N for Nakhun, and K for Kalai. And let me now introduce you to my special guest, Dr. Elanchezhiyan Manikam. He is the director of Tramani campus, professor and head department of microbiology at University of Madras in Chennai. He was many firsts to his name. First one in the world to show the anti-HSV efficacy of DNA vaccines. First in the world to develop an animal model for hepatocellular carcinoma under inducible gene expression system at NIH. First in the world to develop a transgenic animal model for Hep C, and first in the world to show the toxicities of adenovirus vectors in gene therapy. He is recipient of Dolph Adams Award for the most cited research paper and a recipient of honorary degree of medicine in alternative medicine. He has published over 74 newspapers. He is a fellow of Antiviral Research Society, Association of Biomedical Scientists, and International Association of Medical and Pharmaceutical Virologists. He is also the chairman of Microbiology Board, University of Madras, and serves as an expert member for the project in University Grant Commission. So, all welcome, Professor Manikamji. We'd like you to give a short presentation and then we will have an interaction on the latest mutations in the country.
Okay.
Can we have the slide, please, Ritu? Kindly go ahead.
So I can go ahead.
Yes.
Good afternoon, everybody. it is a very great pleasure for me to be part of this uh, presentation uh, through the Med Talks by Dr. Agarwal. I thank you, sir, for inviting me. The topic is going to be viral mutation. I just covered because the SARS coronavirus is the topic of topic of the day everywhere. I looked at a few mutations and variants that we encounter through SARS coronavirus epidemic and pandemic. So, just a few introductory remarks, like to introduce the topic to you. It's an enveloped virus, and it's a single stranded positive sense RNA virus. It has got the genome, which is the very largest, and it ranges from 26 to 32 kilobases. And the important tricky protein within this virus is a spike protein, and that is the one causing a lot of this pandemic very badly. And it consists of two regions, uh, spike one S1 and S2, and the S1 is responsible for the absorption of the virus, whereas the S2 helps in the fusion part of the virus to the cells. And spike protein binds with the receptor, which is very well known, the angiotensin converting enzyme ACE2, and that leads to the endocytosis and translocation of the virus into the cell. And next, please. And this is a genomic organization of this virus. So it has got several non-structural proteins and structural proteins, and you have this large chunk of DNA, RNA, that is PP1A and PP1AB. The two-thirds of the genome encodes for two large polyproteins, that is the PP1A and PP1B. And this portion is cleaved into 16 non-structural proteins. And the last one-third of the virus, the genome, encodes for structural and accessory proteins. Next, please. And this is to show you the same genomic organization. It has got the ORF region, the ORF1A and ORF1B. The ORF1A encodes for the non-structural proteins 1 to 10, and the ORF1B encodes for the non-structural proteins 12 to 16. And after that, you have all the structural proteins. Next slide, please. And as I told you, the spike protein helps the virus, getting fused to the target cell, and the spike protein helps the virus attaching to the ACE2 receptor and fusion of the virus, and it takes care of the entry. And the mutation in the spike protein causes the emergence of newer variant coronaviruses, including SARS coronavirus 2, which is a pandemic nowadays. So the SARS coronavirus keeps on mutating and forming several quasi-species or variant viruses. That's what I'm going to touch a little bit. And this is how the fusion takes place. Next slide, please. And this is the life cycle of this virus. So the virus attaches to the ACE receptor, and then it transcribes, and it for each transcript for a protein. So you have spike protein and several other proteins, and then it gets translated with the help of ribosomes, and then the assembly takes place, and the virus buds out. So there is a simple way, simpler of life cycle of this virus. Next slide, please. And coming to the variants, so this is what causing all these problems these days. The quite a few variants that we had so far, and but the emergence of this variants is due to hyper mutation of this virus, and the hyper mutation happens in this virus because they have a poor proofreading system, especially the RNA-dependent RNA polymerase, and because of that, it is prone for causing a lot of mutation, and this mutation causes several variants. The one variant is a cluster 5. In the cluster 5 is referred as delta FE1 spike. It had several mutations, and it's like 69 to 70 delta HV mutation. So there are like then Y 45 453F, um, and like I 692 to V. So the mutation. So M changes into I. So so there are quite a few variation happens, but this variation it keeps on changes. The other next slide, please. Next slide. So we have several other lineages like B 1.117, and the it was first detected in October 2020. Like it is also like called as like VUI 2020-12/1. They also have they cause lot of problems, and the real problem with these mutants are all there is a change in there is a mutation in the E484 2K mutation, and this strain showed increased transferability and increased lethality. And almost on the similar lines, you had B11207, B11317, different geographical origin emergence. Next slide, please. And several others like lineage B1 429, which is a USA strain, and lineage B1525, which is a UK strain. They all had different mutations. Like in the UK strain, there was a mutation in the E484 E into a K mutation. And then we have the lineage P, the Japanese strain, and it has got a mutation of N501 nucleotide to Y, and E to 484K. So these mutations are seen in the Japanese strain. Next slide. Next. And this led to quite a few missense mutations, like the very popular one, the D614 to G mutation. In this mutation, the glycine is replaced by aspartic acid at position of 614, and it is a very popular mutation and cause of lot of problems. And next please. And we have the V484K mutation. There are N501Y mutation. Next, please. and there are quite a few other mutations like S477 to G or S477 to N mutation, or you can have a P681 to H mutation. Next, please. So, what I covered so far is like there are several of these mutations. Is this the mutation going to be the last mutation? No, it cannot be, because it is a constant, um, um, like mutation, the constant process and spontaneous process. So it keep on mutating, and as of now, the variants whatever we have and the mutation what we have is kind of manageable, different from what we had about a year ago. But however, we cannot exclude, rule out second wave, because second wave we have to be cautious, and wearing we are we are overcoming this infection because of we wearing the mask, social distancing, and hand washing, and we may have to continue doing it until the whole virus is wipes away. With that, I stop here. I'll take some questions, sir.
So my first thing is that we have a virus. This virus has a spike protein and a non-spike protein. And in the non-spike protein, in the first ORF1A and ORF1B, which has 16 non-structural proteins. Out of that non-structural protein, which is in the later part of the ORF1B, that there is a non-structural protein 14, which is a part of RDRP, and which is responsible for the proofreading mechanism, and which can hamper by misuse of many antivirals which act at the level of RDRP inhibitors. So my question is that, do you think that any mutation in the NSP14 will make the virus like a flu, and the virus will lose its proofreading mechanism altogether? Can this happen?
That's what I would imagine, sir. The mechanism is many times very simple. Like uh, whenever the RP, it is a very poor proofreader. So obviously, like there is a mutation there. Definitely, it will. I would assume that it can keep on making flu-like situations.
So what I'm trying to say is, let us imagine from the devil's mind that what does the virus wants? The virus would like to mutate and not being identified by the proofreading mechanism. Therefore, it will ask for a mutation in NSP14, that's one. Virus will like more and more of deletion mutations so that auto-correction cannot take place. If there is a mutation, he would like auto-correction should not take place. And because of 6970 deletion, HV deletion, delta HV in Mings, it became an established virus in UK strain, 6970 along with 144. Maybe 242, 244 deletion in South Africa is also significant. And all the deletions are taking place in the N-terminal domain. So therefore, virus would like to enter into a N-terminal domain mutations as much as possible, so that it ends up into a deletion so that it cannot be recorrected. Then if the virus wants to mutate, he would like to ease out entry into the ACE receptor. For that, it will like to have mutations in RBM more than the RBD portion. Therefore, this 484, 447, all these mutations will come. So it will like to mutate as much as possible and take control of RBD and RBM. It will also like to take control of the fusion part, S1, S2 fusion cleavage, which is 679. Therefore, 680, I'm eight. This is what is that number? I'm just taking a second. And just take a second. And that is, yes, 679. But 681 mutation is already there. So it will like to mutate nearer to 679. If that's so, the more and more we make vaccines and antibodies, there will be a war between the vaccine and the virus. The virus will like to move towards more and more mutations towards advantage virus versus advantage a vaccine. It's like a tennis game occurring between the that who wins, intelligence of virus and the intelligence of human being. And if the mutations are going to occur, why are we so much worried about the mutations? Mutations will keep on occurring. Mutations will keep on occurring. Mutations are still occurring in flu virus. Every year we take a flu vaccine and it reduces your mortality, and that's it. That our vaccines, say every time there is a new variant, you will have to take a new vaccine, and that's it. Why are
Yes,
so much fuss on mutations? Every day, preprint studies, people non-pre-reviewed, every state is coming out with a virus. Is it a vaccine war? Is it a vaccine war who are trying to come forward and say that my vaccine is better, my vaccine is better, my vaccine is better? Why should we be so much concerned about mutations? Will keep on occurring. Mutations have to occur. Mutations have to occur, and more mutations are likely to occur. Even 6970 UK virus is not stable. It will like to acquire, it is already acquiring more mutation. It is already acquiring 484 mutation in New York. It will like to have more and more mutations. So why so much fuss about mutations? That's my question.
but in my view, like mutation are there is going to be there. Like even though like we give this vaccine and all, but I think there will be still certain areas which are conserved, and and which may, with the hope that it may offer some level of protection, and some level of antibody production may happen, which may check to a certain extent, but if at all, it is not going to offer a complete protection. And I think that may be the reason behind this vaccine war. But I agree with you, like whether it will be going to be like a flu shot that we take every year, or that we have to wait and see. I think.
And do you think that like a flu vaccine is an inactivated killed vaccine? And once you know that the virus is going to mutate again and again, again and again, though I did not take Covaxin, do you feel that inactivated killed vaccine, which is a whole vaccine, the whole virus, may be better because it's a virus is going to mutate again and again. In that particular situation, you are producing antibodies against each part of the virus, and that gives you a better immunity than only a spike protein.
I would, yes, certainly, like having a whole viral vaccine is the best way, uh, and because it will definitely going to produce antibodies against all components of the virus, and including the spike, whatever that was there in the original construct.
And do you
Um
do you think that China today has said They will be doing rectal swab of every visitor coming to China to early detect the virus. Do you think that oral virus would have been quicker, better and give a better immunity? Why do you think that from a microbiology point of view, how
Yeah.
does an injectable virus and a oral virus differ? Oral
Um,
vaccine, sorry, not oral virus, oral vaccine.
Oral vaccine.
Why
I
shouldn't from microbiology point of view, why shouldn't a oral vaccine be better in corona when we know that it is present in the stool? That means it might be producing IgA antibodies. Why can't I produce a oral vaccine and produce IgA antibodies from the oral cavity, which also inhibits the nasal IgA? I mean, people are talking about nasal vaccine. Why shouldn't oral vaccine work? Is there a basis for that?
Yeah, I think there are some previous work I came across, like vaccination through the anal route. It induces for sure it induces a very good mucosal immunity at the gut as well as other mucosal area. But how far it is superior than take it orally, that I am not sure. But had I read somewhere like previous work, like it was very potent way to immunize some people. I heard like that.
Why I'm asking this question is because do you think that, like polio, that some of the viruses present in sewage virus will, if taken by a contaminated water, will provide some immunity? Theoretical question. This is a lateral thinking. This is not a.
Um, yeah, like if we get it from water source getting immunized, obviously it can happen, but the number of organisms that is going to be number of uh, I mean organisms in water source may be minimal, may not be enough to induce.
What is
Uh,
the situation of corona now in Chennai?
it is decreasing, sir. It is a decreasing trend only. We hear about neighboring states like uh, Kerala and other places. It is increasing, but so far we did not see any spike or any
And what
increase.
is the virus running in Tamil Nadu? Any special mutation or it is same as D614G?
I say I hear only the regular one, the 6-1.
Regular virus, the wild virus. So let's
Yes,
pick
sir.
up one or two questions before we end. Let's have Balram Saryam. What is your question? Divya says, I am 57, hypothyroid, diabetic. And what is your question? Can I take Covishield vaccine? Yes, go ahead with Covishield vaccine, though your CRP is 2.32. So I will rather give you aspirin and statin on the day of Covishield vaccine. Divya is the same question. Yes, go ahead. You go ahead, Saryamji.
Okay, sir. Okay.
I'm taking Amlos 2.5 milligram for BP. Pulse, should I take it on alternate days? No. Anuji, blood pressure medicine either are taken daily or not taken. If you feel that you are overdosing yourself, then you can ask your doctors to stop the medicine. Uh, yes, if anybody else is there. So if nobody is there, I would like Professor to summarize 5 points in the interest of the people. 5 points in microbiology, what everybody should know. So, Dr. Manikan, can you give 5 take-home messages for the public to know about the virus mutations?
See, viruses, they are they there is a constant fight pursuit between humans and the microorganisms. So in this pursuit, normally our immune system kind of keep a pressure on these microorganisms, and the microorganisms, they find a way to escape this pressure. So one of the mechanisms is the mutation. Mutation is the easiest way by which they can keep on changing their phase, so that the immune system goes obsolete. And this is not only happening with coronavirus. There are several other viruses like HIV does it, flu does it. Many RNA viruses, they does it. But the mutation that happened with coronavirus is a kind of eye opener for all of us. Like there are so many other viruses which are still silent, and this is like a spontaneous mutation. I still believe it is maybe a spontaneous mutation, not a man-made mutation. And this keeps on occurring, and we and nature, because mother nature, takes care of everybody. And I think and for everybody have a space also. So, and this corona situation may be there for a while, and it may go away. That's my suggestion.
So thank you very much for giving a detailed view of mutations. जो लोग इस प्रोग्राम को देख रहे हैं, याद रखिए म्यूटेशंस आ रहे हैं, आते रहेंगे, लड़ाई जादू रहेगी। जितना आप वायरस पे प्रेशर करोगे वो म्यूटेट कर जाएगा। उसके पास में 32 बेस पेयर्स हैं, 32,000 से ज़्यादा उसके पास अमाइनो एसिड्स हैं, 1200 से ज़्यादा उसके स्पाइक प्रोटीन्स में अमाइनो एसिड्स हैं। एक भी अमाइनो एसिड अगर उसने एक्सचेंज कर लिया तो उसकी क्वालिटी बदल जाएगी। वायरस चाहेगा कि मैं हमेशा रहूं, वो क्रॉनिसिटी की तरफ जाएगा। वो चाहेगा मैं हेपेटाइटिस सी की तरह हमेशा बॉडी में रहूं। वो कभी नहीं चाहेगा कि मैं आपको मारूं। वो हमेशा ये चाहेगा कि मेरे को एंटीबॉडीज ना मारे, मेरे को मोनोक्लोनल एंटीबॉडीज ना मारे, मेरे को पॉलीक्लोनल एंटीबॉडीज ना मारे, मेरे को दवाइयां ना मारे, मेरे को वैक्सीन ना मारे। तो वो इस तरीके से चाहेगा मेरी बॉडी में, जैसे कि मैं बॉडी का माइक्रोबायोम बन जाऊं, कि बॉडी में रहूं और थोड़ा 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 अंदर अंदर से बॉडी को खाता रहूं। It will ultimately like to become a low grade chronic inflammatory virus in the body. लेकिन हम नहीं चाहते, क्योंकि हमें लॉन्गेविटी चाहिए और हम बुढ़ापे में जवानी की मौत चाहते हैं, ना कि जवानी में बुढ़ापे की मौत। उस सिचुएशन के अंदर में हमें लो ग्रेड सिस्टमिक इन्फ्लेमेशन अपना रोक के रखना है। That's all for today. Let me thank Dr. Alan to be here with us. We will be back tomorrow 7 p.m. with another show. Keep watching Med Talk with Dr. K.K. Agarwal. Thank you, Elanchezhiyan.
Thank you. Thank you, Doctor.